Welcome to Chemistry 322, Medicinal Chemistry and Pharmacology. In our first lecture, we'll learn how to navigate the course materials and cover class expectations. Then we can dive into some basic definitions, and a mini review of organic chemistry will be coming up in Lecture 2. So how do chemistry and biology come together to create therapeutic agents? In this course, we're going to explore how chemical structure determines the biological actions of drugs, or how medicines are discovered, how they work in the body, and how we design these new therapeutic agents. This is a highly integrative course. We're going to blend concepts from organic chemistry, biochemistry, physiology, and disease biology, so that by the end of the term, you'll understand not just what drugs do, but why they work on a molecular level. So let's start out by defining medicinal chemistry and pharmacology. Medicinal chemistry is the science of designing and improving drug molecules by understanding how chemical structure controls biological function. Many fields of science are involved in this process, including the ones we've already talked about. Pharmacology, on the other hand, explains how drugs produce their effects, how the body handles them, and how these processes determine therapeutic outcomes and safety. It has two major components to it. Pharmacodynamics, which focuses on what the drug does to the body. You'll see this labeled in this class as PD for short. And the second is pharmacokinetics, which focuses on what the body does to the drug. This is labeled PK for short. So our opening unit is going to introduce the core principles that connect chemistry to drug action in the body. Week 1 will focus on foundations and some review of organic chemistry. And then in week 2, we're going to get into more of the pharmacokinetics, absorption and distribution of drugs in the body. And in week 3, looking at metabolism and excretion. During unit 2, we'll look at the drug discovery and development process in a little bit more detail, looking at things like structure-activity relationship and how clinical trials are actually run. And we'll look at more of this molecular design in week 5 and some advanced pharmacodynamics in week 6. During unit 3, we're going to focus a little bit more on modern medicine and its application to actual disease states. And so we'll spend time looking at the cardiovascular system and some metabolic drugs. We'll look at neuropharmacology through the lens of Alzheimer's disease during week 8. Talk a little bit more about anti-infective medicines in week 9. And put this all together in week 10, looking at toxicology and some other modern advances to therapeutics, including some biologics and RNA therapies and gene therapy. So overall, the goal of this course is to develop a chemical and mechanistic understanding of how drugs are designed, how they interact with the body through our pharmacodynamics and pharmacokinetics, and connecting these drug mechanisms to their therapeutic uses. By the end of the course, you should be able to analyze some modern therapeutic approaches and interpret case studies using chemical and biological reasoning. So this course is fully asynchronous, meaning that you can complete your weekly modules on your own schedule. However, it's not self-paced. You will have weekly deadlines that you're expected to stay on track with. So here's what each week should look like. First of all, this is a 4-credit class, which means that there are 4 hours of lecture content that will be required per week. So you really want to schedule these times just like you would for an in-person class. Most students are going to find the best success by setting up 2 2-hour blocks of time to watch your lecture videos, or maybe you would rather have 4 1-hour blocks of time per week. Or you could even choose daily 30 to 45 minute sessions, right? But you want to schedule those into the week. Don't try to block them out and do all 4 hours of lecture in one day. So in addition to having your course lectures, you're going to have homework assignments and weekly quizzes or bi-weekly quizzes. And so we'll have some short quiz question sets that will follow each lecture to help you practice the material. I would recommend doing these right after you watch the lecture so that you gain some practice with the content while it's fresh in your mind. And then you'll have some homework assignments that are more in depth that will help you solidify those core concepts. And so your homework should take between probably 2 to 4 hours of your time each week. 4 is probably on the heavy side, maybe more towards when you have a class assignment that you're doing, maybe with some of the other students in the class. So assignments have to be completed by the weekly deadlines. Late work is marked down 5% for each day late to a maximum of 50%. So you can always earn at least 50% credit on assignments. Your late work must be submitted by Friday of week 10 to receive any partial credit, right? So that is the final deadline, but please don't wait to turn in homework. You want to stay up with the class. The homework is going to reinforce concepts and help you follow the content. So for your midterms, there will be 2 midterm exams given during the term. These are in week 4 and week 7 of the term. These are open notebook, but these will be timed exams. So you do need to prepare for them. Look over your lecture notes before you go in and take these quizzes. You have to do them in one setting under that time constraint. The final exam is going to be cumulative and it has to be proctored. So it's closed note, closed book. So you do need to learn content from this course for that exam. If you're local, I will offer 2 times during finals week where I will proctor the exam on site at WU, and you would have these 2 choices to be able to come in and take that exam. Alternatively, you can have the exam proctored at another university or a library setting. That would be fine as well. And there is a proctor approval form that is available on Canvas. And it must be approved no later than the end of week 8 in this term if you're going to have the final exam proctored off-site. So some time commitment recommendations for the class. You want to definitely have 4 hours blocked out to watch your video lectures each week. And 1 to 2 hours for the homework will be an average for each week. Plus you want to have preparation time for taking those quizzes and reviewing and studying. So I would recommend another 30 to 60 minutes on those. So ultimately you should be spending about 6 to 8 hours each week on this class. So I would recommend treating this course like an in-person science class. So even though the lectures are recorded, it does follow a weekly rhythm, and if you fall behind, it often makes you struggle to catch up. And this is because concepts that we'll be doing in here really build on one another. So I've got some helpful strategies to help you do an online class if you're new to these. Again, you want to schedule your lecture watching time in advance. Don't wait till the end of the week. You want to block out regular times in your calendar to watch the lectures, whether that's daily, whether that's 4 times a week, or whether you want 2 2-hour blocks. Set that time aside and do your work. Take notes as if you were in a classroom. You can pause, rewind, and make diagrams in your notes. You can download the lecture notes and put them on your tablet or print them out, and you can take notes directly in there when you're watching the lectures. This is especially important for things like the pharmacokinetics or dynamic curves that we might be looking at, for structure-activity relationship discussions, and other mechanistic descriptions in the class. You'll also want to engage with your homework soon after watching the lectures. In fact, I would recommend that you go in before you start watching the lectures and look at the homework assignments each week, right? This is going to let you know what to pay attention for when you're watching the lectures, and really looking at some of the key points in the lectures. I'm going to be testing you on the things that I think are important. So you'll want to, open those assignments up, maybe even print them out if they're more essay-based questions, and be ready to try to find those answers as you're going through the lectures. And then reach out to me and ask questions when you need clarification or you're struggling on a problem, right? Even though our class is asynchronous and online, I'm not online all the time, right? I'm at WU, so you can contact me. You could come in person and talk to me, or we can meet on Zoom, especially if you're distant, but we can meet kind of face to face and have a conversation if you need a little bit of help. Sometimes even just 5 minutes will help clarify a situation. So please feel free to email for an appointment and we can set up a time to meet. All right, that takes us to our first lecture. Right, so in this first lecture, we really want to talk about the scope of medicinal chemistry and pharmacology. So by the end of today's lecture, you should be able to define some new terms, like what a drug is, what medicinal chemistry means, and what are the basis of pharmacology, and explain how they're related to one another. You'll also want to be able to distinguish between chemical names, generic names, and brand names, and describe why each is used. And then you'll be able to describe the major stages of drug discovery and the development pipeline, and explain why drug development is a lengthy, costly, and high-risk project. We'll start looking more at organic chemistry and do a mini review in Lecture 2. So that last learning objective here is actually going to be covered in Lecture 2. So let's start out with a definition. What is a drug? Well, a drug is a chemical compound that interacts with a biological system to produce a measurable biological response. An effective pharmaceutical agent is going to be designed to achieve a desired therapeutic effect while minimizing harm to the patient. So ideal drugs are going to have high efficacy, low toxicity or unwanted side effects. They have to be convenient to administer, or patients aren't going to have good resilience taking them, right? If it hurts to give it to yourself or is difficult to plan around, or you have to take it a lot, people aren't going to do that. It also has to be a drug compound that has sufficient bioavailability to be able to reach the target. Once it gets into your body, it's got to go to the right place and function on the drug target. And it also has to be able to be manufactured reliably, efficiently, and at a reasonable cost to make it a good drug. Right? Those are the economic realities to making a drug. All right, so as we said, a medicine is a pharmaceutical agent that has a desired biological effect on the human body or some other living system. Whereas a poison is any agent that's capable of producing a deleterious response in a biological system. And I'm bringing this up early because it's often only the dose that determines whether a chemical compound is going to behave as a pharmaceutical agent or a poison. So even water can act as a poison if enough of it is consumed in a short time frame. Right? So dose really is going to determine whether you have an effective drug compound or a poison to the body. So we said the overall focus of our course this term is going to be on medicinal chemistry, and we've already defined that now. So it really requires connecting the structure of the compound with its inherent properties to the function of the drug molecule in the body. This is going to involve interactions with the drug and the drug target, and this will be a central focus of many of our lectures coming up. So during drug development, there's a focus on the potency of the drug, how biologically active is it, the selectivity of the drug for its desired target, right? We don't want a lot of those off-target toxic byproduct reactions inside the body. We also want to evaluate stability of the compound within the body and the safety of the medication. So we're going to explore all of these different aspects of medicinal chemistry, right? Connecting that structure, property, and function inside the body. And then we'll focus a lot on those two pillars of pharmacology, the pharmacodynamics, what the drug does to the body, and the pharmacokinetics, what the body is doing to the drug. So before we dive in too deeply into the process of designing a drug, let's become a little bit more familiar with how drugs are named, right? And their naming conventions. So really there's 3 types of naming that you can have for a compound. The first is based on the chemical structure, right? And that's going to be the chemical name based on the IUPAC naming system. Honestly, this is rarely used clinically. The names get quite robust and painful the longer the compound or bigger the compound gets, right? So if we're looking at Tylenol over here on the side, here's the chemical structure. The chemical name for Tylenol is N4-hydroxyphenylacetamide, right? So that's already a mouthful, and this is a fairly small compound that has only a few functional groups in it. So when we have our mini organic chemistry review, we'll focus more on looking at these functional groups again and refamiliarizing you with those. But for our class, we're not really going to look at this really in detail at any great extent. Okay, we're going to focus more on the generic naming system in this class. This is international naming for drug compounds. It's coordinated by bodies like the World Health Organization or the WHO and the United States Adopted Name Program for Pharmaceuticals called USAN. And these systems are going to help ensure clarity with those generic names. They have universal identifiers like acetaminophen. So this really refers to the compound. And then the brand names are made by the companies that are making the drug. They're going to be specific for the company name of the drug, like Tylenol would be the brand name. It contains the active ingredient then, which is the generic name for the compound, acetaminophen. So also note that the brand names are indicated with an R with a circle around it so that you can identify brand names very quickly. They're always posted in the upper right-hand corner of the drug name. And in addition to having the noted drug, in the case of Tylenol, the active ingredient, again, is this acetaminophen. It can have special elements that wouldn't be in a generic brand. So you've got a generic brand shown down here, only indicated by the generic naming on that compound. So the Tylenol itself might have different binders or inactive ingredients, and it may have some special formulation for the delivery of the drug that is not in the generic. However, the generic brand has to meet strict guidelines that are set by our Food and Drug Administration so that they have relatively the same activity as the name brand product. So let's talk a little bit more about generic drugs and generic names, right? So the purpose of generic names are really designed to provide a standardized, universally recognized system for identifying pharmaceutical agents. So the generic names, again, they're established through these organizations like the World Health Organization, and they're constructed with 3 key components. They all have a prefix, a stem, and a suffix. So the prefix is going to be a unique syllable chosen by the company that's easy to pronounce and distinct from other drug compounds, especially other drug compounds in the same class. And then the stem or a substem is going to be a specific syllable which conveys information about the drug's class, its target or its mechanism of action. For example, the term statin is a stem for cholesterol-lowering agents. So all of the different cholesterol-lowering agents that are approved on the market contain statin in their generic name, right? But you might have like atorvastatin would be one of those, right? The atorvo would be the prefix used with that particular drug version of a statin drug that is cholesterol lowering. And then oftentimes there will also be a suffix that's a universal marker for the drug family. For example, MAB is used for monoclonal antibodies. The suffix, though, refers to the structural class, but it doesn't necessarily refer to the target. So you can have a lot of different types of monoclonal antibodies that are used for different specific disease states. And at the end of their drug naming, they'll have that MAB present there so that you know it's a monoclonal antibody. But the actual stem is going to help refer you to what the indication is going to be for that drug or what disease it's going to be used for. So generic naming is very useful, and it will be the most useful structure that we'll have for identifying drugs in this class. We'll probably stick primarily to the generic naming and learn about those ones rather than the brand names. Okay, so this is an example of a generic naming scheme. So the drug compound is metoprolol, and its structure is shown here. And you can see that the breakdown of this, the prefix for this drug is the meto, and this is chosen to differentiate the drug from others in the same class. It often will hint at the chemical structure or its substituents, and then the prolol in this case is the stem, and this is identifying this drug class as a beta adrenergic blocker or a beta blocker. You don't know about these drug classes yet, but as we go through the class, you'll be learning more and more about different types of drugs and drug classes, especially in that last unit of the class where we're going to be looking at specific conditions that are being treated, such as cardiovascular disease or maybe neuropharmacological agents used to treat things like Alzheimer's disease. And then the suffix in the case of this drug is the OL ending, and you may remember this from organic chemistry or maybe not, but the OL ending means that there is an alcohol functional group in this compound. So this one is the alcohol, and you can see the alcohol functional group up here in this compound, which must be important for the biological activity of this type of compound. So this slide highlights the key principles that help guide the naming of generic drugs, and this is to emphasize the safety, the global consistency, and the clarity of the drugs. So generic names are really designed with patient safety as the top priority. You want to minimize the risk that you have medication errors taken by the patient, right? To avoid confusing or similar sounding names. You don't want patients to get confused and take the wrong drug, thinking they're taking a different one. They also have to promote global consistency, allowing the drugs to be recognized and understood across different countries and different healthcare systems. And finally, generic names are constructed to really maximize clarity. And this is why they use those standardized stems that help immediately convey information about a drug's function or class, such as the term VIR as antiviral agents. This will help clinicians and scientists and lay people as they become more familiar with them to help quickly infer a drug's role based on its name alone. Or whereas the brand or proprietary naming, it has a major purpose for marketing and consumer recognition. The pharmaceutical companies want to create something that's very unique and recognizable so that patients want to take that brand name drug instead of the generic. Companies do have specific guidelines around making brand names. They can't sound like other drugs. Again, you don't want to make people get confused when they are taking multiple drug compounds. It has to be a very unique name. They also do not allow them to make any misleading claims based on the name or somehow imply superiority in their naming strategy. Like Novo Rapid was not approved because it made it sound like it was going to work more rapidly than other generic forms of the drug. So they dropped the rapid and they ended up calling it Novolog. Companies, again, also want the name to be memorable and culturally acceptable. So some examples of name brands would be Advil, Motrin, and Equate. All of these different name brands contain the active ingredient ibuprofen. And again, that profen stem in the generic name is referring to the nonsteroidal anti-inflammatory class of molecules. So ultimately, there can be some differences between the brand name and generic drugs, but usually these are really small and tightly regulated. Generic drugs are required by the U.S. Food and Drug Administration to be therapeutically equivalent to their brand name counterparts. So what that means is they have to have the same active ingredient, they have to be at the same strength and dosage form. So if it's a tablet in the name brand, it has to be a tablet in the generic, and they have to have the same amounts. They have to have the same route of administration, whether that's oral, IV, or topical. They have to also have the same intended use and the same clinical effects and safety profiles for most patients. So that's an important caveat there. So what's different then? Even though the active drug is the same, some differences are allowed between the brand name and the generics. So inactive ingredients can differ between the brand name and the generic brand. The fillers, the binders, the dyes, coatings, and preservatives, they can affect things like taste and color. They might affect shelf life and tolerability, which can be important for patients that may have allergies or sensitivities. They can also be different in their appearance, the size, the shape, the color of the tablets, or the imprint can be different. This can matter for medication adherence sometimes. If patients are given a generic when they've been taking a name brand drug, they may not recognize the tablet and wonder if they've been given the right drug. And then absorption range or bioequivalence window can be a little bit different and have some variation with the brand name product. But generics have to fall within 80% to 125% of the brand's blood concentration under standardized testing. This may seem like a really wide window, but this range actually reflects the normal human variability between patients that have different abilities to metabolize and absorb the drug when they're taking it. So even like a brand name drug that's taken by two different people, they may have a 80 to 125% range of variability of processing that drug in their body. So that's the same range that you would see then required between generic and brand name drug. So for certain medications, even small changes can be noticeable. So this can be true for drugs that have a very narrow therapeutic index. So we haven't defined therapeutic index yet, but this is a ratio that compares the concentration of a drug at which it becomes toxic to the concentration at which it's effective. So if you have a narrow therapeutic index, that means that you only have a little bit of wiggle room between having an effective dose and having a toxic dose. Examples of drugs that do have narrow therapeutic indices include things like warfarin, a blood thinning drug, levothyroxine, lithium, and certain anti-seizure medications. So for these types of drugs that have narrow therapeutic indices, switching between a brand name and a generic or between two different generic drugs may require monitoring of the patient more closely to make sure that they don't have a negative reaction when they switch. So some clinicians prefer that patients stay on a consistent formulation when they're taking drugs that have narrow therapeutic indices, or if they have patient-specific factors, like maybe they have some allergies or sensitivities to some inactive ingredients, or maybe GI conditions that would affect absorption or things of that. So age can often make a difference. Is a patient a pediatric patient or a geriatric patient? That can affect your ability to absorb different drug compounds and give you different effects then between the brand name and the generic options. The bottom line, though, is that generic drugs are as safe and effective as the brand name drug for most patients. So generics have to meet strict FDA standards for equivalence. So let's talk a little bit about the drug pipeline, and then we'll close out this first lecture. So how long does it take on average to find a drug candidate and develop it so that it's actually clinically useful? Well, not surprisingly, this can actually take decades. The drug pipeline can be very long, right? So first you have to identify a biological target of a disease. What do you want to target inside the body to help treat a disease? Then you have to do lead discovery and optimization of that lead. And this can require molecular modifications of your initial lead. The organic chemist may design different functional groups to test. This can help bioavailability or reduce toxicity, things like this. The organic chemist then has to actually design and synthesize these different compounds, which can take quite a long time. And then you do the biological testing, and this can be an iterative process that goes on again and again until you get something that is binding really nicely to that identified target. So if you have a candidate that passes the lead discovery and optimization, it's going to go into drug candidate development. And the first step is preclinical studies. These can still take years to do these. These need to be done in animal models, and they're looking at safety and bioavailability, the pharmacokinetics, and those pharmacodynamics, so the efficacy of the drug as well. So usually starting in mouse model or rat model, and then moving to maybe some in vitro systems or into higher animals to look at those preclinical studies of safety. If it passes the preclinical studies, then the drug companies can apply to the FDA to do clinical trials in human populations. And if it gets approved by the FDA, then it would go into clinical trial testing and ultimately into becoming an approved drug. So this process is very long and laborious and has many, many steps. We'll break down these steps just a little bit and see what each step really requires. So we talked about that preclinical testing in animals. So really you have multiple things that you're looking at in these preclinical tests. You're looking at the pharmacokinetics and the pharmacodynamics. So you're looking at things like how well the drug is absorbed, distributed, metabolized, and excreted, and the exposure versus the effect of the drug. You want to look at toxicology as well. Are there toxic side effects to the drug, and are these acute? Do these have repeat dose toxicity? Like it's fine one time, but if you repeat the dose, does the toxicity increase? You want to usually have at least 2 different species that you're testing the drug in. Maybe one a rodent population and one a non-rodent population. And then you want to look at the safety pharmacology as well, especially looking at key organ systems. Is it cardiotoxic? Does it affect lungs or the central nervous system or other organ systems in the body? Is there any genetic toxicology? Like, does this cause mutations in DNA? Is it carcinogenic? Will it cause cancer? Is it sensitive to pregnancy? Right? You don't want to, all of a sudden be giving new drugs to pregnant women and have them have bad effects on the forming fetus. How do you make and deliver the drug reliably? Is the formulation actually feasible to do and bring to market? That's more of an economic question for actually being able to make that drug. Okay, so if it passes the animal testing, then the company can apply as a new investigational drug to the FDA to get clearance to start clinical trials in humans, right? So they are going to submit all of their preclinical safety, toxicological, and pharmacological data. They actually have to show that they have high manufacturing quality, that their drug compounds can be produced, and that they're clean and safe for human consumption, so that they don't contain any byproducts or mixed things in there that could cause contamination. And then they also have to come up with a clinical protocol for the phase 1 clinical trials. So they need to identify who's going to be taking the drug, what dose, safety monitoring, and the stopping rules, like if they have an adverse reaction in the patient population that they're testing on. Once the IND becomes effective, then the company can start clinical trials, or the FDA can place a clinical hold until issues are addressed. Like if they see some problem in the protocol, they can say, no, you can't move into phase 1 clinical trials. Or even if they're in phase 1 clinical trials, the FDA is providing oversight and monitoring. So if there's a problem, they can put a pause on the clinical trials straight away. So what do the clinical trials actually look like? They're divided into 3 phases. So in that first trial, our phase 1 trial, this is conducted in humans to focus predominantly on safety. So it involves small groups, maybe 20 to 100 people, and they're usually healthy volunteers or patients that receive the drug at increasing doses, and they're only evaluating tolerability at this point, the side effects, and some of the basic pharmacokinetics. They're not actually evaluating efficacy at this stage, so they're not actually trying to treat any disease state in phase 1 clinical trials. So phase 1 trials will establish the safe dose range for the further studies. Phase 2 trials then are going to start testing whether the drug actually works in patient populations and try to define the dose better. So typically you have 100 to 300 patients in a phase 2 trial, so a little bit larger scale than the phase 1, and you have more focus on the actual efficacy of the drug for treating a disease state. If the phase 2s come back positive and you don't have any safety issues, it can move into a phase 3 trial where you're having much higher populations and trying to refine the dose of the drug compound for biological activity while avoiding any toxicity issues with the drug. So if you have successful clinical trials, the drug company can then apply for a new drug application or a BLA, which is a biologics license application, and they would submit this to the FDA for review. Their submission would have to have the clinical efficacy and safety data from their preclinical trials all the way through the phase 3 clinical trials. They have to have manufacturing controls and validation to make sure that they can safely produce the drug at a very high quality, no contaminants and things during the drug manufacturing process. They would develop a proposed label, what the indications are, what disease states they're trying to approve the drug for, and any warnings or side effects need to be delineated as well. And then any risk management plans the company is making if needed with regards to the drug. The FDA will then follow up the review process by doing facility inspections to make sure that they can produce the compound the way they say they can, and they have quality control measures set up for evaluating the quality of their drug production. The FDA also has advisory committee meetings for some drug compounds, not all of them, and then negotiations on labeling, and then post-approval monitoring has to occur as well. So that post-marketing surveillance, or phase 4 monitoring as it's called, is going to help monitor the long-term safety and effectiveness in the real world when all patients can be accessing this drug compound now for treatment strategies. So you have to have this ongoing pharmacovigilance going on because sometimes you don't see adverse reactions in the clinical trials. So there has to be an adverse event reporting system set up for the drug so that patients can report back in any adverse events that they have while taking the drug. These have to be followed up and investigated when there are adverse events to see if they're real from the drug or if it was something else that just happened at the same time. And so you'd have registries, observational studies that could follow. Additional required trials might be assessed by the FDA. And then you would have any label updates or risk evaluation or mitigation strategies if needed. And that really brings us to the end of lecture 1. So in summary, we've looked at what drugs are as chemical compounds designed to interact with biological systems to produce responses. We defined medicinal chemistry, which focuses on the structure, property, and function of drug molecules, while the pharmacology explains the drug effects and the drug fate within the body. We talked about naming conventions, focusing really on the generic naming that provides information quickly to scientists, clinicians, and patients. And we looked at the drug discovery process and found that it is very long, expensive, and high risk. So it may take screening of 5,000 to 10,000 different compounds to find one lead compound that comes out of that whole mess. And then drugs will fall out in the process along the way, either at lead development stage, you've got a number of compounds falling out, and then at each stage past that, with the biological testing, there'll be drugs that aren't bioavailable enough to move forward into clinical trials. And then at the clinical trial stage, maybe the drug's not effective in the human population, or maybe there's too much toxicity in the human population, and the drug won't make it through the clinical trials. It'll fail at 1, 2, or 3. It can even fail after it's been approved in that post-monitoring stage, and it might be pulled back off of the market. So really, getting a drug to market can cost a pharmaceutical company over $1 billion to get it to the stage where they've actually developed and marketed a single drug lead. So it's very expensive to develop a new drug and get it to market. All right, we'll stop there for today. And next time we'll get into a little mini organic chemistry review.